Welcome to The Explainer. Today, we're going to totally flip the script on one of the most misunderstood words in the dictionary, fat. For decades, lipids have been treated like the ultimate bad guy, blamed for pretty much every health issue out there. But in this explainer, we're stripping away that stigma. We're going to look at the sheer biological brilliance of these molecules, because honestly, without them, life as we know it simply wouldn't exist. To make sense of all this, we've broken it down into 5 key areas. We'll hit the basics of the misunderstood macronutrient, explore nature's energy batteries, map out the shipping fleet, examine the protective wall of our cells, and finally, decode their role as chemical messengers. Okay, let's dive right in. Have you ever wondered why our bodies prefer to hoard fat? I mean, if carbs are our quick go-to fuel, why not just store a massive amount of those instead? It's a fantastic question, and it really challenges how we think about human biology. The secret is all in the sheer chemical efficiency of the lipid molecule, and how its physical shape basically dictates exactly what it does. So, part 1, the misunderstood macronutrient, saturated versus unsaturated. To get why lipids are so efficient, we've got to look at fatty acids at their absolute most basic level. And spoiler alert, not all fatty acids are created equal. It all comes down to the degree of unsaturation, which is really just a fancy way of saying there are double bonds in their carbon chain. This drastically alters their physical state. Animal fat? It's predominantly saturated. That means its chains are super straight, they pack tightly together, and that makes it solid at room temp. Plant fat, on the flip side, is generally more unsaturated. And this side-by-side comparison perfectly captures that real-world difference. On the left, you've got animal meat loaded with palmitate, a fully saturated fatty acid with this rigid, straight structure. But over on the right, look at the canola seeds. The oleate in canola oil is unsaturated. See that little double line in the oleate structure? That double bond puts a literal kink in the molecule. It physically stops the lipids from packing too tightly, and that is exactly why canola oil stays liquid. All right, moving to part 2, nature's energy batteries, triacylglycerols. So how exactly does the body bundle these fatty acids for storage? Well, get a load of this number. When your body completely oxidizes triglycerides, which, is our stored fat, it pumps out roughly 38 kilojoules of energy per gram. Compare that to the oxidation of glycogen, our carb storage. That gives us only about 6 kilojoules per gram. So gram for gram, fat gives you over 6 times the energy. To put that in perspective, a moderately obese person carries enough stored energy in their adipose tissue to survive for weeks. Our glycogen stores, they'd be wiped out in less than a single day. Check out how elegant this triacylglycerol structure is. It's so beautifully simple. Just a backbone of glycerol locked onto 3 fatty acid chains. And because these molecules are entirely nonpolar and incredibly hydrophobic, they absolutely refuse to mix with water. What does that mean for us? It means they can pack tightly into our fat cells without bringing any useless water weight along for the ride. It's basically the most weight-efficient biological battery imaginable. Part 3, the shipping fleet, transporting lipids. Now, this incredible density creates a bit of a logistical nightmare. Think about it. How do you move completely water-hating batteries through our entirely watery bloodstream? Well, digesting fats from your last meal is this crazy multi-step orchestration. First, dietary triglycerides are emulsified in the intestine by bile acids, kind of like how dish soap breaks up grease in a frying pan. Then, lipases step in. They act like tiny molecular scissors, snipping them into free fatty acids so they can cross the intestinal wall. Once inside, they're reassembled and packed into these specialized shipping containers called chylomicrons. The sheer engineering of this transport vehicle is wild. This chylomicron is the largest lipoprotein particle we have. Notice how it builds this perfectly safe vessel. The outside is a monolayer of phospholipids and free cholesterol that happily interacts with the watery blood, while the inside safely hides massive amounts of those hydrophobic triglycerides. It's essentially a biological submarine navigating your circulation to deliver fuel to your tissues. But your liver doesn't just sit around waiting for your diet to supply fat. It can actually synthesize its own triglycerides straight from carbs and proteins. It bundles them into a different type of shipping container called a very low-density lipoprotein, or VLDL. Think of the liver as your body's central distribution hub, constantly shipping VLDLs out into the bloodstream to guarantee every single tissue gets the energy it needs. So, what happens when you suddenly need that energy? Let's say your blood sugar tanks, or you're suddenly facing a threat. Boom! Hormones like glucagon or epinephrine hit the alarm. They bind to receptors on your fat cells and trigger this rapid-fire chemical cascade. That cascade activates lipases right inside the fat cell, slicing open those stored triglycerides and flooding your bloodstream with free fatty acids, completely ready to be burned for fuel. Okay, part 4. The protective wall. Cell membranes. So, we know how lipids store and ship energy, but what they do structurally is just as mind-blowing. Behold the fluid mosaic model. Your cells are not locked away behind a rigid, solid brick walls. Actually, scratch that, they're encased in this dynamic, flowing ocean of glycerophospholipids. These lipids arrange themselves into a double layer that creates a flawless hydrophobic barrier. It shields the delicate internal machinery of the cell from the chaos outside, but it's fluid enough to be embedded with proteins that act as doors, windows, and sensory antennas. And prepare to be a little shocked by this stat. Between 25 and 40% of the lipids in this plasma membrane are actually cholesterol. Yep, the exact same molecule we've been conditioned to fear is literally vital for your survival. Its rigid 4-ring steroid structure wedges itself right between the other lipids. It acts as this perfect buffer, keeping the membrane fluid enough to function, but stable enough not to just fall apart. Within this flowing ocean, lipids organized into these specialized microdomains we call lipid rafts. Picture them as highly organized floating islands made of sphingolipids and cholesterol. And they aren't just floating around for fun. They serve as absolutely critical meeting platforms. By clumping together, they make sure specific cellular receptors and signaling proteins can actually find each other and link up in the vast open space of the membrane. Now, to really grasp how vital these specific membrane lipids are, we just have to look at what happens when the system breaks down. Tay-Sachs disease is a devastating condition, and it's caused by a defect in just a single enzyme, hexosaminidase A. Its one and only job is to remove a tiny sugar molecule from a specific lipid called a ganglioside. When that enzyme fails, those gangliosides build up in the brain and spleen, causing severe developmental issues. It's a stark reminder that every single lipid molecule has a very precise, non-negotiable job. Finally, part 5, chemical messengers, lipids in cell signaling. Moving way beyond structure and energy, it turns out lipids are some of the most potent chemical messengers we have. Let's look at cholesterol one more time. Not only is it stabilizing our cells, it is the ultimate biological starting material. Your body uses it to synthesize crucial steroid hormones, everything from stress hormones like glucocorticoids to sex hormones like estrogens and androgens. It's the precursor for the bile acids we talked about earlier. And get this, when UV light from the sun hits your skin, it literally converts a cholesterol derivative straight into vitamin D, the sunshine vitamin. Then we've got a totally different class of lipid messengers called eicosanoids. These guys are derived from arachidonate, a specific polyunsaturated fatty acid in our cell membranes. Eicosanoids are incredibly cool because they are so phenomenally potent, but they only stick around for a few minutes. They act locally, right where they're produced, serving as high-affinity messengers that kick off everything from your immune response and inflammation to pain signaling. And to make these eicosanoids, our bodies rely on enzymes called cyclooxygenases, or COX enzymes. COX-1 pumps out lipids that protect your stomach lining, while COX-2 creates the lipids that specifically trigger inflammation, pain, and fever. So why does this matter to you? Because next time you have a splitting headache and pop an NSAID like ibuprofen, you're taking a drug that's literally designed to block these exact lipid signaling pathways. You're cutting the pain message off right at the source. So, we've traveled from the basic structure of a simple fatty acid all the way to the molecular mechanics of pain relief. We've seen how these unbelievable molecules build the literal walls of our cells, pack away the dense energy that keeps us alive, and orchestrate the chemical signals that dictate our health. So what does this all mean? I'll leave you with this. If lipids are the master architects of our biology, our most efficient survival batteries, and our most vital chemical messengers, is it finally time we stop treating them as the enemy and start respecting them as the foundation of life? Thanks for exploring this with me today. Keep learning and keep questioning.